For the next 45 minutes or so, I would like you to join me on a journey in time and space to look at the microbiome and how it may affect us, and especially our physiology and metabolism. I usually like to start with this cartoon that one of our co-workers, Anna Helena's, made, because I like to view the microbiome as a big family. We have some species that have a close relationship, maybe they are brothers and sisters, and others are cousins or second cousins. Just like in the microbiome, we have some strains that are very closely related, whereas others from other genera are quite distant. That said, it means that not necessarily two siblings have very close similar functions or interests. You can have a similar function or interest or similar job as one of your cousins. The same is going on in the microbiome, that some species that are closely related may have very different functions, whereas the same function may be inherited by or developed by a more relative species. So let's start this journey in time and move back some 3,000 years ago. These are from an old Chinese book of medicine, where they described some couple of thousand years ago the yellow soup. What it meant was if you had gastroenteritis or gastrointestinal infections, it could be cured by a yellow soup composed by feces from a healthy individual. This was then refined. You took the feces from a child, and then in the 400 AD, they start to actually ferment this feces to make it more efficacious. So almost 1,500 years ago, you used the microbiome to cure diseases. The next concept is the Old Greek. This is Hippocrates. He was he's named to be the inventor or shaping modern medicine. And in one of his works, he's attributed with the words, "Let food be thy medicine." So I think it's very appropriate for being at the Food Institute to have yellow soup and then Hippocrates, "Let food be thy medicine." Because we know that the microbiome is very much affected by the food we eat. And that's this combination I will come back to during this lecture. Let us fast forward a millennia or so into the 1600s, because neither the old Greeks or the old Chinese, they didn't know what they were doing. They just knew that they took feces and it would actually cure a disease, or you could have food to cure a disease or prevent a disease. But they didn't know the active components. It was not until Antonie van Leeuwenhoek invented the modern microscope in the 1600s and used that to study not only cells from plants and from animals, but also to look what he had in his oral cavity. This is a picture of what he saw in his oral cavity. He also looked in his feces and could, for the first time, depict bacteria, but he called them animalcules. And he could see that some of them had motility, others were large, others were cockeye, and some were spirochetes in their shape. He could also then show that when you had an episode of diarrhea, the composition changed. So he was the first person to really illustrate a dysbiotic microbiome. If we move another 200 years forward in time, we have Ilya Mechnikov, who went out on the countryside in Bulgaria, where he saw that some of the peasants there, they seemed to live a long and healthy life, and they were eating a lot of yogurt. And in yogurt, you know that you had lactobacilli. So he came up with the terminology probiotics for life from that observation. He later on went on to get a Nobel Prize for his role in studying immunology and immunity. So he was father of phagocytosis. About the same time, Louis Pasteur, who is then known for his pasteurization, were discussing that he thought that life is not possible without any microbes. It turned out that he was wrong, because he spurred scientists like the German Nuttenthal to start thinking about developing germ-free mice that have never, or germ-free animals. He actually worked with guinea pigs, animals that never were exposed to any microbes. Nuttenthal had some problems technologically, but his work was followed up by James Reniers, who was a young student. He was 19 years when he went to the dean of Notre Dame University. Can you imagine a 19-year-old person coming to the dean of DTU saying, I need a lot of money because I want to build the first environment for germ-free animals. I have no prior experience, but I'm pretty sure I can do it. So he got resources to do this, and 8 years later, he managed to produce the first germ-free rats and keep them over generations. This was highlighted in a Milwaukee Chronicle in 1936. And it's quite amazing, but already at that time, they asked the same question as we do today, whether humanity would be better off or worse off without any wicked little bugs to contend with. And this started the golden ages of the first golden ages of studying the normal gut microbiota. It lasted out until around 1980. But then you have compared and contrasted almost all forms of germ-free mice or germ-free rats with rats and mice with a normal microbiota. So you know that the villi was different, the immune system was different, the appetite was different, etc. But there was not much more to explore until we had a next leap of technology. This was the event of using knockout mice, genetic manipulation of bacteria to study mechanisms. It was, I think, above anything else, probably the democratization of sequencing, for example. So you don't have to culture the bacteria in the gut, but you can sequence them to see what are the bacteria in a given person. And this allowed then to start to compare and contrast different patients and controls and see that the microbiota differed between many different diseases. And finally, I think it also spurred the public that studying the microbiota could have a big impact on our life, on our daily lives. So I've been fortunate to catch this second wave or second hype of the microbiome research. And the main characters of our research are on this scanning electron micrograph, where we have colored the bacteria in orange. We have the mouse colon mucosa in gray, where you see the microvilli. We have stained the mucus that we produce to protect our cells and also to provide a niche for bacteria in green. And then we have some food particles in purple. So these are the main actors of today's talk. And we like to say that we're studying the gut microbiota. Some call it gut flora. I prefer microbiota because we're not studying flowers. But it's a little bit, we usually study a fecal microbiota sample. And talking about gut microbiota when you're actually studying the fecal microbiota, I know I will interchange this today, but just to give you an example on why I think this is a bit misleading. For me, it's a little bit like studying the Earth from space. And although you know that Antarctica and Sahara are very two different habitats, we can't clump them together. We should probably start to study more the small intestinal microbiota and maybe the upper colonic microbiota, etc. It's technologically challenging, but I think that's something we can do together in the future. Because if we compare again with ecosystems, what we see along the length of the gut, we start in the stomach. It's like a desert. There's not many bacteria that can thrive there. That's one exception, that's Helicobacter pylori. That's why we have this palm tree there. Then when we enter the small intestine, the duodenum, I think that's like a tundra, because the bacteria there are exposed to all the digestive juices coming from the pancreas, the bile from the gallbladder, and you can have bile soap to clean clothes. So this bile is very toxic to most bacteria. So it's a very harsh environment. That's a low biomass. And when we come down to the Scandinavian pine forest in the ileum, that's when we start to see the biomass with some 100 million bacteria per gram content. But for me, I think the colon is probably the most interesting space. It's really where you have the bulk of the bacteria. To me, it's where we have the diversity and most of the metabolic reactions going on. So I like to look at the colon as a very lush rainforest. So if we then take the step back and say people are studying the fecal microbiota, which we claim to be the gut microbiota, you can find basically any disease you can think of. If you Google that disease, and microbiota, you will find a paper describing that there's a difference between this disease and controls when you look at the microbes. But these are just associations. So I think we have to come over that and start to study whether there's a causality, and I will come back to that in the end, where I think this is critical. And in our work, we've been mainly focused on the cardiometabolic diseases, such as obesity, for example, which has increased dramatically over the past couple of decades. We see living in an obesity epidemic, and in the wake of that obesity epidemic, we see increase in type 2 diabetes. This is a map that shows the prevalence of type 2 diabetes, or diabetes actually, but most of it is type 2 diabetes. So you can see that in some of the countries here in the Middle East, almost 20% of the population has already diabetes today. And this is projected to be increasing even more, and especially in low and middle income countries. And unfortunately, individuals living in these countries will probably not afford or have access to the modern treatments. So maybe by understanding if the microbes can affect this disease progression, can we find ways of either prevent or potentially also help in treatment of individuals for these cardiometabolic diseases. So one way of moving beyond associations, it's to use germ-free mice. Germ-free mice, of course, mice are different from humans. I'm fully aware of that, but it's one way to at least start to understand if the microbes can affect a given process. So what we tend to do is we use these plastic bubbles where we have the mice, and it's almost like a space station. In that environment, there's no bacteria. So in order to keep it germ-free, we have to import sterile foods and water and supplies through a port in the back, just like when you get stuff into a space station. Then we can handle the mice through these gloves. We can feed them different kind of food, and we can colonize them with different kind of bacteria or fecal samples for that matter, to see if a given bacteria or a fecal sample can confer a disease. When I started working on this some 20 years ago, that was not so much known. So we wanted to see, is there a difference between amount of body fat in a germ-free mouse and a conventional raised mouse? So as a postdoc together with Jeffrey Gordon, we took germ-free mice and we took normal mice and we put them into a DEXA machine that can estimate the amount of body fat. And what we saw was that the germ-free mice, in absence of any bacteria, although they ate 30% more, had less body fat. If we then took and killed these mice, took the bacteria, entered it into these germ-free mice, and analyzed those mice 2 weeks later, they had normalized the amount of body fat. So when we wrote that paper, I wanted to make it that the germ-free mice were normal, and the colonized mice had more body fat. In hindsight, I think we should have probably done it opposite, that the normal mice are normal, and the germ-free mice are malnourished instead. And what the microbes are doing when you feed them this regular chow diet, it's mainly consists of different fibers. So the bacteria will ferment that fiber and provide energy to the mice, and that's why we have more body fat. And this is sort of the situation we humans have been having up to like maybe 70 years ago. There was a shortage of food, so having microbes helping digesting and giving us energy was probably something evolutionary beneficial. So in the 1980s, it was estimated that about 10% of the energy of an individual living in the UK had this energy coming from microbial fermentation. And even if something has been evolutionary beneficial a couple of, maybe 100 years ago, if you change that environment, it may actually be detrimental. So if you then compare and contrast instead mice that are germ-free and colonized and give them a Western-style diet. This is a diet that mimics what you get at McDonald's or another burger joint. Right? So 40% of the energy comes from sugar, 40% comes from saturated fats, and 20% from protein. We can then see that mice with bacteria gain weight. Germ-free mice do not gain weight to the same extent. This is, they gain about 5% maybe, and these guys gain about 20% in 8 weeks. So we don't know if this actually is true in humans, but Henrik, who's here, and his group will examine that now, I hope, and figure that out to see whether what we see here, if the energy extraction could be important. But even if obesity is a first sign of cardiometabolic unhealth, it's really a moving target. Usually you start with increased obesity, especially abdominal obesity. That increases the risk for metabolic syndrome, to type 2 diabetes, and also to cardiovascular disease, and then later to diabetic complications. So at least I think that maybe the microbes can help with energy digestion, but the most reason why we are getting obese is probably that we take too many calories. But not all obesity are alike. Why do some individuals develop diabetes sooner than others? Can the microbes be modulating the time on this or direction of this journey? So why did we start? Well, we and many others started in the same way. You compare and you contrast a group of healthy controls and a group of individuals, patients with type 2 diabetes. And if we move to 2010, when those studies were usually initiated, one of the hallmarks of microbiome research was that there was no chance of replicate someone else's findings. So it was a big thing when we actually got our paper here got accepted in Nature because we could replicate what this Chinese-Danish group have done. We found the same thing. So the argument for us to get our paper published in Nature was that we could replicate another study, which is quite unique. And what both of these groups found was that in patients with type 2 diabetes, we see fewer butyrate-producing bacteria. Life was good, we moved on, and then, of course, there's another paper showing that a lot of the studies at that time were confounded by metformin usage. Because if you study a patient suffering from a disease, they usually have a treatment. And that treatment can also affect the microbes. In this case, metformin is the most common first-line treatment for diabetes, and metformin per se can affect the microbes. We had control for it in our study, but we were still requested by reviewers to say, are you sure you're studying the disease and not the treatment? So we moved on and we teamed up with a physician at the Wallenberg laboratory to study treatment-naive individuals. These are individuals in the society that they may have diabetes, but they don't know it in that case, so they have no treatment for it. So we built a cohort of some 2,000 individuals eventually by screening the population and selecting those with prediabetes or diabetes and doing a lot of examination on them. And together with Tony Wu, who was a postdoc at the time and now a professor at Fudan University in China, and Valentina Tremaroli, who's our team leader in microbiome research, we sequenced the microbiota of individuals with either normal glucose tolerance test or normal glucose tolerance, people with impaired glucose tolerance, or people with type 2 diabetes, or they had prediabetes based on both impaired fasting glucose and impaired glucose tolerance. And what we could see, that none of them were on diabetes treatment. We saw that the healthy controls in this space of microbiomes were up here, and then you saw a gradual decline on the microbiome when you had patients with type 2 diabetes. So what we had found in the first study was true, but we also started to think maybe we can do an analogy here. Because if you read Anna Karenina, it's from Leo Tolstoy, it opens up with that all happy families are alike, and each unhappy family is unhappy in its own way. What we think is that if we take a microbiome-centric view on this, is that a healthy microbiome is rich in butyrate producers and have a high diversity, so they look relatively the same. But when this microbiome starts to fall apart, for example in diabetes, they will have blooms in many different bacteria, so it will be hard to find a statistical significance for that. So it seems to us that if you eat your fibers, if you eat a diverse diet, you will have a rich microbiome and you will probably be protected. That will probably help protecting against type 2 diabetes. But this is still only associations. So we do our trick with the germ-free mice. And what we do then is that we take individuals from that study that have screen-detected type 2 diabetes, so they didn't know that they had diabetes when they came to us, but we diagnosed them. And we take an individual here, and then we find a healthy control that has the same BMI, same sex, and it has the same age. And we do that for 6 different individuals in each group. We then colonize germ-free mice, and we make a cohort of then some 20 mice that have been colonized by microbiota from patients with diabetes or individuals living with diabetes, and another corresponding group with that have been colonized with normal gut microbiota from controls. And Louise Manneros, who's leading our germ-free efforts, has then been following these mice, and she could see that we colonize them, they gain weight the same way. When we monitor their glucose, it's similar, but when we monitor their insulin that controls the glucose, it's much higher in the mice that were colonized with microbiota from individuals with type 2 diabetes. So I'm not a diabetologist, there's others here that are, but very simplified, if you have a high insulin sensitivity, you need just a little bit of insulin, this blue line, to control the glucose, which is in the green line. What happens then is that you start to get insulin resistant, so you need more insulin to control the glucose, so that's why the blue line is increasing. And then eventually you have a lot of insulin produced, but it's not sufficient, so you have more glucose as well. And eventually the beta cells producing insulin dies, and the glucose goes through the roof, and that mouse or a human would die if they're not treated. So what Louise did, she called those mice that had had a normal glucose and normal insulin, she called them A. If they had normal glucose but high insulin, she called them B. If they had both high glucose and high insulin, they called them C. And if they had low insulin and high glucose, she called them D. And after the experiment, she then could see that the mice colonized with control microbiota, they were mainly in the A and the B group, whereas mice colonized by the microbiota from type 2 diabetes individuals, they were mainly in the C and the D group. So we think that the microbiota can really at least contribute to the glucose regulation in mice in this case. This is data that we haven't then followed up on, but our colleague Max Nudorp, who's also in this microbiome health initiative, I will tell you a little bit more about later. He did a quite fascinating experiment some over 10 years ago. He took individuals with metabolic syndrome, and he performed an insulin clamp on them. And if you have a lean control, they have a very high insulin sensitivity. That's what's measured here. If you take a patient with metabolic syndrome, they have a low insulin sensitivity. If you take the feces from a lean control, put it into this patient, and measure the insulin sensitivity later, it's improved. But if you instead take a patient with metabolic syndrome and give him or her their own feces, it's not improved. So this suggests that the microbiota can also help insulin sensitivity or improve insulin sensitivity in humans. Max repeated it here in another study, but what's a little bit depressing is that it only lasts for about 6 weeks. Then you have to make a second transplantation, and having a fecal transplantation is probably not the nicest to have, nor you also have potential transfer of pathogens, so you may get sick of it. So what are the microbes that seem to be protective? Well, we talked about butyrate-producing bacteria, and the bacterium that stands out the most in our analysis is a bacterium called Faecalibacterium prausnitzi. You can't probably read it here, but it's the top one here on the list. So instead of giving a fecal transplant, we would like to give individuals a capsule with a bacterium that we can control much better. So Tanveer Khan, who's a scientist in the group, he's been really trying to build a strain bank or bacterial bank based on what we find in a healthy individual's feces. So he takes feces, puts on a plate, there you get these colonies, and each colony here stems from one bacteria that grows. The bacteria divides maybe once every 20 minutes. So over a night, the fast-growing E. coli can become somewhere a billion bacteria. So in each dot here, you have identical bacteria. And by streaking them in several rounds, you can improve the purity. So Tanveer wanted to identify a sulfate-reducing bacteria, a bacteria that can take care of sulfate in the gut. And it's called the sulfovibrio piger. So the plate we culture it on gets all black. But what Tanveer found was that in this mixture, it was not only one bacteria, it was actually a second bacteria, actually this Faecalibacterium prausnitzi. So his idea was then that these two probably thrive together. And he could test that by culturing them together. So if you take Faecalibacterium prausnitzi by itself, it grows to a certain extent. But it's increased by some 70% if you instead add its partner bacteria, the sulfovibrio piger. And it's really this cross-feeding. They have a compatible metabolism. So on this specific medium, which is basically on having glucose and a little bit of lactate and sulfate, none of the bacteria can produce butyrate by themselves, but together they can actually produce significant amount of butyrate. So this is all good. Now we have actually found a bacterium that seems to be protective against type 2 diabetes. We found a partner bacteria that make it grow better. But another problem is that these bacteria living in our gut, they don't tolerate oxygen. They are very anaerobic. And you may be able to see that on this slide here, that on the left-hand side, we have the Faecalibacterium prausnitzi, what we culture in a tent deprived of oxygen. But if we have exposed them for 20 minutes of oxygen, the plates are completely pure. So what Tanveer then did was that he developed a little gym to train and exercise these bacteria by giving them a little oxygen at a time, yet keeping them in good shape by giving them some both nutrients and control the redox balance by electric current. And for each subculture here, he increased the oxygen a little bit. So eventually, we have a bacterial strain that is fully trained, and if you expose them for ambient air for 20 minutes, you have some growth. Not as good as anaerobically, but you have then developed this adapted strain. So this then made it possible for us to do a clinical trial. I'm not an MD, but we did it together with Matthias Lorentzon, who has done a lot of first-in-man trials. And then Chen Mei, who's a postdoc in the group and now an assistant professor in Umeå University, analyzed the data. So we had placebo, we had a low dose and a high dose, about 15 individuals in each group. And what we could see was that the high dose of the product increased the levels of disulfovibrio piger in the feces of these individuals, but we couldn't see an increase in Faecalibacterium. And we think this is because there's so much Faecalibacterium in the healthy gut. So giving 10 billion extra doesn't make a difference. So we are really excited about this, and we would like to take this further and see if this product could be protective against cardiometabolic diseases. We are not only interested in who is there, we're also interested in what they are doing. And one big difference between the microbiome and our other organs, because we would like to view the microbiome as an organ on its own right, maybe weighing half a kilo to a kilo, similar size as the liver, composed of different cells. But in contrast to the liver, the cells in the microbiome or the microbiota will change over life. We're born sterile. We have then get a climax of bacteria that approximately when we maybe 20 to 50 years, then we start to lose a little bit of diversity as a population in the microbiota. But we also change on a shorter time span. This could be due to what we eat. If we take any drugs, of course, antibiotics would have a bigger impact, but even metformin, as I mentioned, or statins would have impact on the microbiota. So you can change both the composition, but also then giving substrates to these microbes that give rise to different molecules and metabolites that can actually function as hormones or affect distant tissues. So our proposal is that we actually are host to an endocrine organ within our intestine. And of course, these metabolites could either be beneficial or detrimental. So what we've done now is together with Tony Wu, is to estimate how many metabolites do we find in the plasma of individuals with type 2 diabetes, and how big proportion of them are associated with type 2 diabetes, are produced by the microbiota, or regulated by the microbiota. And what we did find was that 500 metabolites were different between healthy controls and patients or individuals with type 2 diabetes, and about a third of them were microbially modulated. So it's clear that the microbes can produce many different metabolites that can affect our health. And we have put a lot of focus on a metabolite that we were happy to find in this analysis as well. It's called imidazole propionate. And it's a metabolite that the bacteria can produce, but we cannot produce because we are lacking those enzymes. So the bacteria, and we, for that matter, can take histidine, which is an amino acid, metabolize it to uroconate, but whereas we and the bacteria can metabolize it further to glutamate, only bacteria can reduce this double bond to imidazole propionate. So it's a microbially specific metabolite that we know is associated with type 2 diabetes, and we could validate that. So here's a study with some 2,000 people from a study called Metacardis, and Trine, so sitting in the back there, was very much involved in the Danish part of that study. And it's not maybe super impressive, but we see that we have more individuals with a type 2 diabetes or pre-diabetes have higher levels of imidazole propionate in general. But what we think is that if we look at the top quadrant here, this is where you see the main difference between those healthy and those with type 2 diabetes. So we think that perhaps these individuals with type 2 diabetes here may have, that their imidazole propionate may have a contributing factor. Once again, these are just association studies. So we want to move on and test the causality. So here we feed mice with a Western-style diet to make them more prone to cardiometabolic diseases. Half of them are treated with imidazole propionate and half are not. We then perform a glucose tolerance test. So we challenge the mice with glucose, and we take samples from the tail, and we monitor the glucose, and we see that mice that were treated with imidazole propionate, they have an impaired glucose tolerance compared to those that were not treated. This is work done by Hobbe Agrawal, who's a postdoc in the group. But we also want to understand what are the mechanisms involved in this signaling pathways. So what Ara Ko, who's now a professor at Postec in Korea, found was that whereas insulin usually binds to the insulin receptor, leading to signaling through the insulin receptor substrate, and then to AKT phosphorylation, what Ara found was that imidazole propionate can activate a signaling pathway regulated by these MAP kinases, and which eventually leads to you have a abnormal phosphorylation of insulin receptor substrate that prevents the activation and rather targets it to degradation, so you can shut down the signaling pathway. And this was, so you say, well, is this important in humans as well? So looking at samples from livers, so liver biopsies from patients without type 2 diabetes, so these are obese individuals that are in the clinic for bariatric surgery. So we have obese individuals without type 2 diabetes, and we have some with type 2 diabetes. And what we can find is that this P62 phosphorylation is increased in individuals with diabetes. The S6K1 phosphorylation is increased, and the insulin receptor substrate is reduced. So we think that this is a pathway that is not only active in mice or cells, but also in human livers. So for us, this is very exciting, but can it affect how we live, how long we live, and the risk for different cardiovascular events? So in work that we've done together with Stan Hazen's group, led by Antonio Molinaro and Ina Nemeth from Cleveland, we analyzed individuals at baseline. Then we divided them in 4 groups. So the highest group, 25% with the highest levels of imidazole the second highest group of imidazole and then the second lowest and the lowest. So the quartile 1 are the 540 patients with the lowest imidazole, and quartile 4 is with the highest level of imidazole. Then we follow something called MACE. It's called Major Advanced Cardiovascular Events. It's a composite of stroke, myocardial infarction, or death by cardiovascular events. And if you had the high levels of imidazole, you had a worse survival rate over 3 years, or have higher risk of having a MACE compared to those with lower levels of imidazole. And even if we moved further on and looked at survival overall, we saw a similar pattern that individuals with high levels of imidazole had a much higher risk of dying than those with the lowest levels of imidazole. So we do believe that this is a metabolite that can act prognostic, is associated with disease, produced by the microbes, and can confer disease when you treat mice. So what this potentially opens up for is that maybe we should start to think about medicine in a different way. Can we start to, instead of developing medicine to our body, rather drug the bug, so make medicine to treat our microbes? In this case, we would aim to find an inhibitor, like a drug, that would target the enzyme responsible for urocanate reductase to abrogate the production of imidazole. So this is something we hope in the future to be able to deliver on. So since I'm the one between you and the refreshments, I would like to summarize what we discussed so far. That the gut microbiota is for sure altered in many cardiometabolic diseases. We know now that the microbiota is altered before overt disease onset. So individuals with prediabetes, for example, have already an altered microbiota. It's really characterized by a loss of butyrate-producing bacteria. And if we take Max Nudorp's data here, that we know that the microbiota can modulate insulin sensitivity, not only in mice, but also in humans. We think that the development of the next generation probiotics, with, of course, the technological advances such as oxygen tolerance, is a potential view forward to target these diseases. But also, since the microbes can produce many different metabolites, some would be positive, we can add them directly, but others we need to find novel treatment modalities, such as inhibiting the production of enzymes such as urocanate reductase. So now you may wonder, what will I be doing here at DTU? Well, I have the pleasure and privilege to lead together with Tine Rasklicht and with great support from the new team we're developing with Nora Ottman as the center coordinator, and Anna Jackson-Fouché as our administrator, leading the Microbiome Health Initiative, which is a center sponsored by the Novo Nordisk Foundation. So we hope that we can, over the next 4 to 6 years, really try to demonstrate if the microbiome can causally be linked to cardiometabolic diseases in humans. And we also have partners here like Henrik and Trine and Tina, who are very much involved in this Microbiome Health Initiative. And of course, I also like to thank the group in Gothenburg that contributed a lot to the work that I'm presenting here today. So with that, I would like to thank you so much for inviting me to DTU and to the National Food Institute, and see maybe if there's any questions. Thank you very much.